One biological theory of aging is widely known in the lay public as well as among scientists who do not study aging in particular. It has been popular and attractive for decades, probably due to its clarity. It is sometimes called the oxidative stress theory of aging. This theory originates in a paper published by Harman in 1958. It consists in the conjunction of two hypotheses. The first one is that there is a link between the observed effects of radiation on organisms and a possible cause for aging, the natural and avoidable production of free radicals, OH and HO2, by metabolism. Harman writes, viewing this process in the light of present-day free radical and radiation chemistry and of radiobiology, it seems possible that one factor in aging may be related to deleterious side attacks of free radicals, which are normally produced in the course of cellular metabolism on cell constituents. This first hypothesis focuses on free radicals. It both assumes that they are a common effect of radiations and of metabolism, and that they are the common cause of aging and of the effects of radiation. The second hypothesis is called the rate of living theory. According to the rate of living theory, aging is proportional to metabolic activity, so that the concentration of free radicals would increase proportionally with the accumulation of damage due to metabolic activity. This would explain the acceleration of aging. The oxidative stress theory of aging thus explains why aging is ineluctable. It is a direct consequence of respiration and metabolism, i.e., the production of energy in the cells. It explains why caloric restriction seems to have a protective effect, delay aging, and increase lifespan. It also explains the comparison between aging and radiations. It fits nicely with the genetic theory of aging, according to which aging consists in the accumulation of damages in genes, on the condition that we admit that free radicals hit genes and produce mutations. Harman renewed his theory in two later important papers in 1972 and 1978 by applying it to mitochondria. According to this version of his theory, mitochondria are the main responsible for the production of ROS, free radicals, in cells. Of course, there are many other sources of ROS: ultraviolet radiation, inflammation, heat. Mitochondria are organelles, that is, components of the cells. Their function is to produce energy for the cells by making up ATP. But a side effect of this process is the production of short-lived, unstable small molecules, ROS, that would bump into the first bigger molecule they would encounter and disfigure them randomly. Given that there are a lot of molecules in a cell, ROS are likely to meet one very close to the place where they were produced, that is, in mitochondria themselves. The most sensitive target in mitochondria would be their genome. Mitochondria have their own genome, distinct from the genome of the cell, properly speaking. It is a very small genome, and it is not protected like the genome in the nucleus of the cell. By damaging mitochondrial DNA materials, ROS would make them more dysfunctional with time. Given that mitochondria reproduce by division, as bacteria do, a genetically damaged mitochondrion would divide into two genetically damaged mitochondria, so that damages not only impair the functioning of a mitochondrion, but are, so to speak, hereditary and propagates within the cell until it becomes inefficient due to bad energy production. In this theory, a paradox has to be explained. Indeed, mutant mitochondria, that is, the ones that have accumulated damages, are less and less functional, and thereby produce less and less free radicals. How is it that they just don't stop multiplying? Some scientists, including Aubrey de Grey, see the module on the sense theory, proposed the idea that damaged mitochondria are less useful for the cell but fitter than undamaged ones, precisely because they produce less free radicals. For this reason, they multiply until they make a majority of the mitochondrial population in a cell. More recently, some have insisted on so-called redox homeostasis. That is, the idea that ROS are not only useful to the organism, but that there is an optimal balance of the concentration of ROS in cells, and that this balance progressively tilts with time. An aged organism or a tissue damaged by an age-related disease such as atherosclerosis or cancer is supposed to be characterized by an increasing proportion of radical oxygen species, ROS. Theoretically, this is all very fine. But given how short-lived ROS are, it is actually very difficult to measure. The theory has been supported and challenged by mixed evidence since its inception. Some have showed a correlation between levels of mitochondrial ROS and longevity. Others concluded to the absence of correlation or even inverse correlation, suggestive that a higher level of ROS would play a protective role, or that without being causal properly speaking, it would indicate a certain level of damage in mitochondria. Many age-related diseases such as cancer, atherosclerosis, neurodegenerative diseases, arthritis, diabetes have been shown to be associated with a higher concentration of ROS. While there is a general idea that animals with higher metabolic activity live shorter lives, and that caloric restriction delay both aging and age-related diseases in many animals, some remarkable exceptions are an embarrassment to this theory. Typically, birds and some long-lived mammals have very high metabolism and a lot of unrepaired damage, likely due to ROS. Even in more rudimentary organisms without cell replacement, like C. elegans, oxidative stress may increase lifespan. Some have proposed the theory of mitohormesis, that is, the idea that a low level of stress on mitochondria might be beneficial for mitochondria. Nowadays, the theory is far less popular among scientists. Recently, some have explored the role of NAD+, an enzyme or coenzyme that plays a role in redox reactions in cells, jointly with other enzymes. It has been shown that the cell concentration in NAD+ changes with age and suggested that modulating it could impact lifespan. In particular, it would stimulate the activity of sirtuins, enzymes that are involved in genetic repair. Others have focused on sirtuins, but it is clear from the literature that this line of research is declining. A critical review can be found here. Since the early 2000s, this theory has developed in the lay public to the point of justifying a prosperous industry of diet supplementation widely known as antioxidants. It is clear now that such supplementation does not come with clear benefits for health in general.